Okay, so good morning, everybody, and let's continue our discussion of this chapter with the discussion on the concepts on prokaryotic cells. Now, we have been learning that prokaryotic cells and eukaryotic cells are quite different, and the prokaryotic cells are primitive compared to the eukaryotic cells. So, prokaryotic cells have certain features, something unique to these prokaryotic cells only, and they are actually not found in the eukaryotic organisms, right? And these structures have certain unique functions. Even they have certain additional structures that are actually not found in the eukaryotic organisms. Now, yesterday we have started with the basics, and we saw the difference between the prokaryotic and eukaryotic cells. And today let's continue on that part, and let's understand certain additional concepts related to this particular concept. So let me just share the screen with you people. Just a second. Okay. Can you see the screen, students?
Yes, sir. Yes.
Okay. So yesterday we have just started with the basics difference between the prokaryotic cells and the eukaryotic cells, and I hope you remember this, where we talked about the different organelles, right? That is that is part of the prokaryotic cells. For example, we had talked about the various structures like the cell envelope, mesosomes, chromatophores, nucleoid, flagella, pili, fimbriae, but we had not talked about in detail. So today we are going to do that exactly. So the first structures, let's start from the outside, and when we talk about that, it is the structure called cell envelope. Now, this structure is unique to this group of organisms. Why? Because we do not see this cell envelope in other organisms. What is the cell envelope exactly? This is a protective layers, right? Chemically complex protective layer that is made up of mainly three different layers. What are those? One is called glycocalyx, second is called the cell wall, and third it's called the plasma membrane. You all have a pretty good idea on what is the cell wall and what is the cell membrane, or that is called the plasma membrane, or it is also known as the cell membrane, right? You all know about this because the cell membrane is something that we see in all the cases, in all organisms and plants. Cell membrane is present. Cell wall is present in certain cases. For example, in case of the plants we have, in case of the bacteria we have, in case of the fungi we have it. But now there is another structure called glycocalyx. If you see this diagram here, what you see, this outer layer, thick outer layer. Now this part is called glycocalyx, right? This area is called the glycocalyx area, and next to that, what we are going to have is a cell wall, right? This yellow part, and then finally we have this membrane, that is innermost plasma membrane, and inside, inner to that, we have the cytoplasm and the other cell components that we all know about. Now, when we talk about the glycocalyx, now this glycocalyx is the outermost layer of the cell envelope, as we talked about, and its composition and thickness is going to be different across the prokaryotic organisms. That is, across the bacteria, their composition and the thickness is going to be different. Now, depending on whether it is thick or thin, there are different names that is given. So it is called a slime layer. As you can see, if just a second, is there any problem? Just a second.
So joining request.
Joining request. I think I have disabled the this thing. I am not enabled the 1 second. Let's check again. Yeah, it's not at all. Anyway, I'll just send the link once again. Where is that? Okay. Anyway, without waiting room also, people are asking to let me in. How it is possible? Anyway, so let me get back to the concept here. So I was talking about the differences in the cell envelope, right? So here the cell envelope, as I was talking about the glycocalyx, it varies from one group of bacteria to another group of bacteria. And if it is thick, it is called capsule, and if it is thin, it is called, or loose sheath, it is called slime layer. Now, chemically, what they are made up of? This is glycocalyx. Chemically, what are they made up of? Now, there are two structures called the glycolipid. It is one of the chemical structure that you learn in higher class, and the other one is called the glycoprotein. Okay, now this glycolipid and glycoprotein, these two structure, they chemically constitute, okay, this particular glycocalyx. So chemically, two components, glycolipid and glycoprotein. I request all of you to please switch off your microphones. Please switch off your microphones. So the next concept here is going to be the second layer here, that is going to be the cell wall. Now the cell wall is the middle layer of this cell envelope, and it is seen in all the prokaryotic organisms, right? All the bacteria have the cell wall. And what is the role? Role is very simple. It is very well present to provide the shape to the cell, and it provides mechanical support or structural support to the bacteria to prevent it from bursting or collapsing, right? The bacteria is actually exposed to various kinds of solution. It can be hypotonic, it can be hypertonic, and in that situation the cell can shrink or the cell can actually swell up and burst. So to prevent the cell from undergoing these processes, the cells are going to be having with or present with the cell wall. Now in this case, the cell wall is actually made up of polysaccharides, and to be precise, it is called the peptidoglycan. Okay, peptidoglycan. It is also known as murin, right? It is also known as murin. Now this peptidoglycan is made up of, it is basically consisting of different type of sugars. And these sugars, this modification of sugars, there are of two types. One is called NAG and another one is called the NAM. Now NAG stands for N-acetyl glucosamine, and the NAM stand for N-acetylmuramic acid. But anyway, you don't have to worry about this much. If you remember that they are consisting of the peptidoglycans or the murin layers, that's more than enough. In the NEET classes, anyway, we'll talk about these in detail. Now, finally, we're coming to the innermost layer of the cell envelope, and that is going to be the plasma membrane. It is the innermost layer, of course, and it is going to be semi-permeable. It is going to be selectively permitting certain substances to get in, and it will prevent certain substances to get into the cell. So they are basically termed as semi-permeable in nature, and they will interact with the outside, and they will decide what will get in and what will get out of the cell. Now their composition is very much similar to the eukaryotes, and if you remember class 9th concepts, you might know about the composition that is going to be the lipid bilayer along with a number of proteins, the major components. So the proteins with the lipid bilayer is a major component of the plasma membrane. And when we talk about the proteins, again, the proteins are of two types. These two types of proteins are, anybody can tell me what are the two types of proteins involved here? Can anybody tell me what are the two types of proteins in this case? Yes or no?
Sir, phospholipids?
Not exactly. Two types of proteins. I would say based on the position in the cell membrane, we can say. One outside, one in between. Can anybody tell now? Okay, I'll just tell you about that. One is called the surface proteins, that is present on the surface of the membranes, and the second one is called the integral proteins, right, that are present along with the lipid bilayer that forms the channels actually that allows the substances to move through the membrane. So there are two categories of protein that are part of this membrane, and we will talk about in detail. One is called surface protein, and the second one is called the integral proteins. Now, based on the cell envelope, we can classify the bacteria into two categories. Now, based on what? Based on the organization and response to one staining procedure called Gram staining. Okay. Now, this particular technique was actually developed by Christian Gram, and based on his name, we have this, naming called Gram staining techniques. Now, there is a particular stain that is used in this case, and this the primary stain is called, right, there is a primary stain that is called crystal violet, and this crystal violet is actually used for staining the bacterial cell. Now there is another one called counter stain here. Okay, counter stain is also used to basically give the that contrast against this crystal violet, and that is called the basic fission. FUCHSIN, basic fission. So two stains are there, but mainly the primary stain you have to remember that is going to be crystal violet. The procedure again you don't have to worry about, but then you will talk in the later classes. So there are two categories of bacteria based on whether they can retain the crystal violet stain or they cannot. So gram positive is a group of bacteria that take up and retain the gram stain, right? They retain it, they have it. And the other one is called gram negative because they cannot retain the gram stain. Now, as you can see here, the E. coli bacteria, which is one of the most common form of bacteria that we actually learn about, and they are gram negative. They cannot they cannot retain this crystal violet stain. Whereas the Staphylococcus aureus, they are a group of gram positive bacteria. Now, let's understand certain modification membrane structures. And when we talk about membrane structure, the first structure that you understand is going to be called the mesosomes. Now, the mesosomes are, okay, before I go into that, let me share with you people a video that's talk about, let me just move this one to some other place. Yeah, 1 second. Yes, so let me share the computer audio. This video is going to allow you to understand the concept nicely on gram staining.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as murine. Peptidoglycan.
Right. As we talked about peptidoglycan or the murine, as I was talking about, they're consisting of this two structure that you can see here, NAM and NAG. Right. So this is made up of murinic acid, and this is N-acetyl glucosamine. So these two structures, as you can see, they are cross-linking and they are forming this peptidoglycan cross-link. And if you see that in case of the gram-positive bacteria, it is typically a little bit thicker compared to the other case here. So anyway, let's continue.
Glycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram-positive or gram-negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then a counterstain is added, for example, safranin or fuchsine. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. Note that gram-positive bacteria also pick up the pink color of the counterstain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to differences in cell wall structure, which is the chief difference between gram-positive and gram-negative bacteria. The gram stain detects peptidoglycan, and since gram-positive bacteria have a thick multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye for two reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. Although both gram-positive and gram-negative bacteria can be pathogenic, gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall. Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified through gram staining. Acid-fast bacteria and gram-variable bacteria, for example, do not respond to gram staining. Acid-fast bacteria are bacteria whose cell walls retain stains particularly well. Although they aren't closely related to gram-positive bacteria, they can appear purple after the gram stain test. Gram-variable bacteria show a mix of pink and purple cells when stained. If you like this video, like and subscribe. You can also support me by...
Okay, I think you are clear on that. Hello.
Yes, sir.
Yeah.
Yes,
So
sir.
Yes,
yeah.
Yes, sir.
So that's the difference between the gram positive and gram negative bacteria. And when you go to higher education and you actually take up any research in biology or microbiology, this is one test that actually we all should know and you have to do it. Even I remember when I started my research career, I had to start with this technique only. So even I took few classes for the MTech students just to teach them what is the different techniques that is used to actually differentiate between gram staining, gram positive and gram negative bacteria. It's a very common technique that is actually used and you all should know about it. The entire, the technique, the practically how it is being done, that you will learn at a higher level, in a higher class anyway. So let me come back here with the concept on the mesosomes, right? So as we're talking about, these mesosomes are nothing but it is infoldings of the plasma membrane. So if you see now this particular one, right, this is our plasma membrane and this plasma membrane is having this infolding, and this infolding here, now that one is going to be called your mesosome, right? Now it includes the vesicles, tubules and lamellar structures. So what is the function of that? Various functions are that you all know that there is no membrane bound structure, so their functions are kind of dedicated to different structures, and one of that is going to be the mesosome. So here they are involved in cellular formation, involved in the replication of DNA or the chromosomal DNA replication, right? For the distribution of chromosomes to the daughter cells, for the respiration and secretion of certain substances, to increase the surface area for the plasma membrane, to increase the space for enzymatic activities. As you can see, there are a number of functions related to the mesosomes. And if you see these functions, some of these functions are actually performed by different organelles in case of the eukaryotic organisms. So here there is no organelles for that. And for that reason, these are actually performed by the same structure that is going to be called the mesosome here. Now the next structure is called chromatophores. Now I have the name, so this chroma here refers to color, of course. And when we talk about this chromatophores, we are talking about pigment containing membranous infoldings. And these are actually found in, for example, the cyanobacteria, also known as the blue-green algae. You all know about that. Now let's talk about the reason called the nucleoid, right? As you all know, last time I was talking about. Now can you remember DNA is negative or positively charged?
Negative, sir.
Very good. They're negative. Yes, they're negatively charged, right? Can you tell me why they're negatively charged?
Sir, due to the phosphate group present in that.
Excellent, excellent. Due to the phosphate group or phosphate moiety. Now we had talked about that this DNA associate themselves with a certain proteins, and these proteins happen to be positively charged. Now why are they positively charged? Because of the amino acid composition, right? There are certain amino acids and these amino acids will give an overall positive charge to this protein. Now you all know that if you bring a positively charged and a negatively charged biomolecule, they will of course attract each other and they combine, tightly get bound to each other. And the reason, the area in which you have this DNA and the RNA, that is your cellular, please switch off your microphone. So cellular DNA or chromosomal DNA and the proteins are actually bound together. That particular area, that particular reason is called nucleoid. Remember, nucleoid is not a structure, it is not membrane bound, it is not any form of enclosed compartment, but instead it is a region floating in the cytoplasm. So आपको दिख रहा है, if this area, right, this particular area, that is the nucleoid, and you can see now this is a chromosomal DNA that is actually, right, this is what, this is your chromosomal DNA. Now that area is going to be called nuclei. So it is formed of non-membranous naked circular genomic DNA, single chromosome, that is your genetic material, and protein. So many bacteria have single small circular DNA plasmid. I think we had talked about this last time also. These are extra chromosomal, remember. So this plasmid, right, these are extra chromosomal. That is, they are present in addition to the chromosome. That's why they are called extra chromosomal circular DNA. And these give some unique phenotypic characters to the bacteria. For example, as I was talking about, they can survive in a condition with medium containing antibiotics. In a typical condition, see, for example, if I'm having a medium, right, this is a medium, and if this medium contains antibiotic, now what is going to happen is they cannot, here the bacteria cannot survive. But if there is a antibiotic medium and here there is plasmid present, then the bacteria can survive in this condition. So this basically is one of the example. There are different type of plasmid, as I was talking about, that give them different property, and each of this property will allow this bacteria to have certain additional features which is going to be absent, which the other bacteria that lack the plasmid cannot have. So that's why the presence of this structure called plasmid, extra chromosomal, double standard circular DNA, which can basically, by the way, replicate by themselves. They do not depend on anything. So they can, again, right, they can self-replicate. So they are not dependent on anything. They can replicate by themselves. So self-replicating structure, that is called the plasmid. Now the next structure, that is extension of this prokaryotic cells, you all know, are going to be the flagella. Now this flagella structure you have learned in previous groups also, or in symbol, that is called flagellum. This thin filamentous extension has the ability, or basically provides the ability of the bacteria for motility, or to move around. To be motile allows them, the bacteria is going to be able to move because of this structure. Now, depending on the number of flagella, whether they're present or absent, you have different terms. For example, atrichus, amphitrichus, lophotrichus, peritrichus, right? All of these, again, you're going to learn in the later classes. But mainly there are three parts which are going to be the outermost, or this, longest part called the filament, then next structure called hook, and there's a basal structure called the basal body. Now, if you see here, now this OM refers to the outer membrane, and this outer membrane is nothing but the glycocalyx, right? Glycocalyx or the outer membrane. Then you have the peptidoglycan layer, right? PG refers to peptidoglycan layer, which is nothing but the cell membrane. They're going to make up the cell membrane. I hope you remember that. This is cell membrane. And of course the CM, right? Okay, peptidoglycan together. This is going to perform. They have made it separately, but anyway, that is a cell membrane structure. So these structures, this peptidoglycan is actually, I'm sorry, this is not a cell membrane. This is the cell wall. I'm very sorry. And finally here you have to going to have the. Okay, let's just change this a little bit and change the color so that it's better. So here the peptidoglycan is going to be the cell wall basically, and the CM here refers to the cell membrane, right? So in that area, in this particular part, basal body is going to be stuck, right? From which you are going to have the formation or attachment of this entire flagella, and then you are going to have the hook and then the filament. So this hook and this basal body is going to be fixing this entire structure. Hook is going to allow this entire structure to basically rotate and move around. So the filament has the longest portion and extends outside or from the cell surface to the outside. Now there are a number of this small bristle-like structure that are the pili or the fimbriae. Now these are surface structure, but they have, remember, no role in the motility. Unka motility or movement में कुछ भी काम नहीं होता है. So what role do they have? Now, when we talk about the pili, these are tubular structures made up of special protein, and this special protein is called pilin. Now, another structure that is actually involved, which are similar to that of pilin, these are called fimbriae. Now these are bristle-like structure, and they actually come out of the cell, and they actually help in attachment, or they allow the bacteria to get stuck, attached to certain substratum, which can be rocks in the stream, and to the host tissue in case they are parasitic in nature, in which is the most of the cases they are parasitic. So they will allow the bacteria to remain attached with the host organism. Now just a little bit about this, the pili or pilus we can call it. Now there is one a special type called the sex pili. Now these are involved in a process that is actually unique to the prokaryotic cells, and this is called the conjugation. Okay, these are called conjugation. Now, what do you mean by conjugation? This is one type of what you call it's a type of horizontal gene transfer. Okay, ये जो horizontal gene transfer is simply called HGT. Now, what is that? I'll just explain briefly. Now, there are two types of gene transfer. One is called vertical gene transfer. Vertical gene transfer means from the parent to the offspring. To if the parents are transferring the gene or chromosome or genetic material to the offspring, then it is called vertical gene transfer. लेकिन अगर वो नहीं है, if it is between two organisms, right? Let's say two bacteria, then it is called horizontal gene transfer, or not from the parents, right? If the gene transfer is not from the parents to the offspring, then it is called horizontal gene transfer or HGT. There are different types. For example, you have the conjugation, then you have the transduction, right? Different types are there. But when we say conjugation, there are involvement of two bacteria, and these two bacteria are connected through this structure called sex pili. And this sex pilus connects these two bacteria, and through the sex pilus, right, the gene gets transferred. Mainly the let's say the plasmid gets transferred from one donor to the recipient strain. So there is one called the one second. So there are something called the donor strain and the recipient strain. So what happens? These two strains are connected through this sex pili, and from the donor strain, the genetic material gets transferred to the recipient strain. So that is the process of conjugation, which actually is one type of the horizontal gene transfer that we see in case of the prokaryotic organisms. Let's wind up with one or two more concepts. Now, ribosomes here, they're associated with the plasma membrane in the prokaryotes, and they're having a size of 15 nm by 20 nm. nm stands for nanometers. And you talk about prokaryotic or eukaryotic, they are consisting of always two subunit, and one subunit is going to be larger subunit, and the other one is going to be the smaller subunit. So when we say larger subunit, in this case, it is going to be 50s, and smaller subunit is called the 30s. So ideally, 50 and 30s together form the ribosome. So it should be ideally 80s, right? 50 plus 30 is going to be 80s if you add, but that is not the case. It is not 80s. Instead, it is going to be 70s in case of prokaryotic cells. Now, S stand for Schwarzschild unit. Schwarzschild unit. It is a unit to represent the sedimentation coefficient, which is a measure of density and the size. So when they're separate, these two have two different Schwarzschild unit, that is 50 and 30. But when they are as a unit, right, function unit, they are not going to be 80s, but instead it is only of 70s. Now remember that 70s only. And if you can see here, right, 50s, 30s, but together it is 70s. Similarly, in case of eukaryotes, it is going to be 60s and 40s, but together they are not 100s. Instead, it is only 80s. So that's the difference with the prokaryotic and eukaryotic ribosomes. Now, when we talk about the functionality, what happens? These ribosomes, in case of they're involved with the protein synthesis that you know from class 9, right? So what happens? Translation is a process of protein synthesis. Now, in case of prokaryotes, a number of these ribosomes, they're attached to the mRNA. So what you see here, this is the mRNA structure, right? And this is the larger subunit, and this is the smaller subunits. They're associated, and a number of these associate within the mRNA, and they form a continuous chain-like structure, which is called polysome or polyribosome, right? And this you see only in case of the prokaryotes or the bacteria. So what is that? A chain of several ribosomes being attached to that of the mRNA, and that, dear student, is called the polysome or polyribosome, which is again associated with the process of translation or protein synthesis. Now there are another structure called inclusion bodies, and these are non-membranous, and they store resolved food materials, and they are available freely in the prokaryotic cells. And for example, you have the phosphate granules, right? Synophysin granules, glycogen granules, gas vacuoles, mainly for storage of the materials. And gas vacuoles are also seen in case of blue-green and purple-green photosynthetic bacteria. Now, as you can see, these are the inclusion bodies, right? Mainly for the storage functions. So that, my dear students, completes our understanding and discussion on the prokaryotic cells. Now, your job is very simple. Read it up once. कुछ doubt है, कुछ problem है, please ask. But